So today's video is on the cell cycle, and the cell cycle is just the cycle of a cell where it grows and replicates. And the cell cycle includes two phases, which is interphase, which is the section where the cell performs its usual activities, and this takes up most of the cell cycle, and mitosis, which is our cellular division, which is proportionally a very small section of the cell cycle. And this diagram here represents the cell cycle, and we'll be looking into each of these sections in a minute. So our first section is in interphase, and this is G1. So you can see that here, it's the first section of the cell cycle in interphase. And G1 stands for the growth stage 1 or gap 1. And in G1, the proteins necessary for new cellular organelles are produced, because we know that for our cells to replicate, there needs to be twice as many organelles, so each cell has its own set of organelles, so proteins necessary for this have to be produced. And also the volume of cytoplasm increases, because there needs to be enough cytoplasm for our two daughter cells to be full identical copies of our original cell. And then we have S phase, and this is also in interphase. And S phase stands for DNA synthesis stage. And by the name, you can probably tell that this is the stage where DNA replicates semi-conservatively. And semi-conservative replication is when our two original strands act as templates for the new strands of DNA to be formed. So our new DNA helix is made of one original strand and one new strand. So S phase is the synthesis of DNA, it's DNA replication. And then we have G2, which is the last part of interphase. And G2 stands for growth stage 2 or gap 2. And in G2, our organelles grow and divide using the proteins and nutrients that we obtained in G1. And also our energy stores of ATP are increased. And this is because when the cell exits mitosis at the end of the cycle, it wants to restart its cellular processes very quickly. So it needs to have this store of ATP to start processes quickly again after it exits mitosis. And then M phase is part of the cell cycle, but this is not in interphase. M phase is mitosis. And this is our nuclear division, PMAT, which is we're going to be going into another time, and also cytokinesis, which is the actual cellular division. And just to reiterate, no DNA replication actually occurs in our M phase in mitosis, because DNA replication occurs in S phase, in the interphase. And lastly, there is G0, but G0 isn't technically part of every cell and their cycle. G0 stands for, well, stop. It is when the cell exits the cell cycle, so the cell no longer carries on this process to go and divide again. And this can be permanent or temporary. So if this cell exits the cell cycle permanently, this could be a nerve cell. It's not going to want to replicate again. So it goes into cell cycle arrest. It doesn't go on to replicate anymore. And we have a diagram representation of this. Now, this is a diagram of showing our cell mass and our DNA mass during our cell cycle over a period of time. So this shows how our cellular mass changes and our DNA mass changes over the course of the cell cycle. Now, the black box represents G1. And you can see this because our cytoplasm is increasing in G1, meaning our cellular mass is going to be increasing. But in G1, no change to the DNA is made, so our DNA mass stays the same. The orange box represents our S phase, because our cell does continue to grow, as we can see here. Our cellular mass is still increasing, but in S phase, our DNA is replicated, so our DNA mass also increases. And then the blue box represents G2, and this is because our organelles replicate, so we are increasing our cell mass, but there is also no change to the DNA like in G1, so our DNA mass stays the same. And lastly, we have the yellow box, which is M phase, our mitosis. And if you can probably see better on this cycle, the cell divides and returns to its original mass. So it goes all the way back to the start, dropping quickly because we've lost the extra mass that we've produced to make this other cell. So it goes back to its original mass. And also this happens with the DNA mass as well, because we're halving it, getting rid of that other cell and splitting it into two. And then we're going to be looking at checkpoints, and these are very, very important in our cell cycle and in the body. And checkpoints are at the end of G1 and G2, and it is in the M phase as well. And checkpoints make sure that everything in the cell is correct and that the cell should go on to replicate in mitosis. And so we'll go into what that means now. So our G1 checkpoint, so this is at the end of our G1 phase. Our G1 checkpoint checks to make sure that the right nutrients are present, and it checks for this so that it can allow the cell to duplicate all the organelles correctly. Because if we go into G2, where we want to replicate our organelles, if not all the right nutrients are present, our organelles will not be made correctly or effectively enough for the new cells to survive properly. And it also checks for growth factors, so whether the cell grows large enough to be able to split into two cells, and also checks for DNA damage. So this is whether the cell should go on to replicate its DNA. So if a cell has damaged DNA, we do not want to replicate this cell, because we don't want to replicate the damaged DNA and gain more mutations or damaged cells. And then there is G2 checkpoint, and this is at the end of G2. And our G2 checkpoint checks for cell size. So this is to make sure that there is enough cytoplasm for two cells. So when it goes into mitosis, it has enough cytoplasm to split to make two identical cells to the original cell. And it also checks DNA replication. So it sees if all of the DNA in S phase has been replicated and if it's been replicated correctly. Because if not all the DNA has been replicated, then there will be some missing chromosomes from one of our daughter cells, and if it's not replicated correctly, we will have mutations in our daughter cells. And then there's the metaphase checkpoint, and this is in mitosis. So this is our metaphase stage in mitosis, and you can see that here. And our metaphase checkpoints check for the attachment of chromosomes to spindles. So this is to make sure that all of the spindle fibres have attached to the centromeres of all of our chromosomes, to make sure that equal numbers of chromosomes are pulled to each pole. Because if one spindle isn't attached to a chromosome, then that chromosome will not be able to be split. It will not be able to be pulled to the opposite poles, so there will be more or less chromosomes in one of the daughter cells, and this can cause mutations that we do not want. And so now we've looked at the checkpoints. What happens if the checkpoints detect a fault in our cell? Well, firstly, the checkpoints will cause the cell cycle to halt to see if the problem will be solved. And so it halts the cell cycle before progressing to make sure that the cell can deal with this fault. But that's all good, but what if the problem isn't fixed and it cannot go on to progress in the cell cycle because the checkpoint is halting it to stop, because there is a fault? Well, if the problem is to do with growth and nutrients, the cell will just enter G0 phase. So it will exit the cell cycle, it will stop replicating. And if the problem is to do with damaged DNA, this is a much bigger problem because this could go on to something, to lead to something much worse. And so the body programs its cell death, and this is apoptosis. And we'll go on to see why the body programs this apoptosis of damaged DNA. Well, the importance of checkpoints. So if the checkpoints don't work properly, they cannot detect mistakes. And this is very important because if they cannot detect mistakes, they could not detect that some DNA is damaged, meaning they'll go on to replicate this damaged DNA, and this will increase number of mutated cells. So if it does not detect in the G1 checkpoint, for example, that the DNA is damaged, it will go on to replicate this damaged DNA and create more mutated cells that could potentially get more and more dangerously mutated with each cell division. And it can also lead to uncontrolled division because the checkpoints cannot regulate what cells are ready or should divide. And this uncontrolled division can lead to cancer. And you can inherit genes for faulty checkpoints. And this is why some people are more likely to develop cancer, because their checkpoints cannot regulate what cells they divide. So now going back to our damaged DNA and why our cell programs this cell death. Now, if we have damaged DNA, we now know that it can lead to uncontrolled division of mutated cells, which can lead to cancer. So that is why if there is damaged DNA that does not get fixed, our body will program this cell death, because this is very, very dangerous